Good afternoon, everyone. My name is Dr. Isabella Pedrosa Pacheco. I am the group leader of the Laboratory for Functional Immunology here at the University of Oxford, and most recently I became the program leader of the Mexico Cancer Initiative, which aims to understand immune responses in CRC patients both in the UK and Mexico to develop better vaccine strategies. As you know, colorectal cancer is amongst the most commonly diagnosed cancer worldwide. According to the World Health Organization and the Global Cancer Observatory, approximately 2 million new cases were diagnosed in 2020, which is equivalent to one case every 15 seconds. With incidence rates rising in some regions due to lifestyle and dietary changes, this number is expected to increase. When diagnosed at an advanced stage, the relapse rate following surgical removal of the primary tumor with curative intent is as high as 60%, highlighting the urgent need for improved treatment strategies that go beyond what we have at the moment. In Mexico, CRC cases are expected to double by 2040, and with approximately 20% of cases being observed in young individuals, especially in Hospital General. The factors are involving diet and lifestyle. This includes alcohol consumption, high sugary diets, physical inactivity, and obesity. And this is really important in the context of Mexico, as it has one of the highest rates of childhood obesity worldwide, and this is something that we need to tackle as a public health concern. To this end, since actually 2016, the government has really improved and made a significant effort into combat the obesity epidemic through strategies to reduce also obesity by prevention. This involves front of pack food labeling, taxes on sugary drinks, and high calorie non-essential foods. In the context of diagnosis as well, through the incorporation of small studies with the use of fecal immunochemical tests that have been discussed, and also routine endoscopies and treatment worldwide with the use of immune checkpoint blockade treatments such as CTLA-4 and PD-1, which are efficient in a subset of patients with advanced stages of CRC. Now, understanding how the immune system responds to cancer at every stage of the disease can really help us improve the treatments that we have at the moment. In our lab, we are very interested in a specific biological processes which are undergoing in the secondary lymphoid organs, and this is actually germinal center structures. As most of you might know, germinal centers are temporary lymphoid structures that are formed upon vaccination and infection. And these are hubs where B cells undergo somatic hypermutation and affinity maturation to provide antibodies of high quality against the pathogens that are being presented at the moment. And we and others have shown that these tightly controlled processes are happening through molecular and cellular mechanisms. And by using murine models and also in vitro studies with human patient cells, we have shown that the role for CD40 follicular helpers, which you can see here in the middle in the graph, that provides cognate receptor ligand interactions to the B cells to undergo somatic hypermutation and acquire affinity maturation. These processes are really important in germinal centers. But also we have shown that there's high levels of cellular regulation occurring outside these pockets. And these include the role of natural killer cells, CD4 regulatory T cells, and most recently we have shown regulatory B cells that control the differentiation and the formation of pre-T follicular helper cells that go into the germinal centers. And this is really important in the context of HIV vaccinations, where we know we're tackling for an HIV vaccination strategy, the development of proline neutralizing antibodies. And we know that these BNAbs are restricted by tolerance mechanisms, this work that we had done in the last 10 years. So understanding the biology and modulating this through a vaccine strategy is really important for efficacious vaccines. Now, while critical in mounting antibody responses and very critical in the context of infectious disease vaccinations. B cells have been mostly overlooked in the immuno-oncology context. And this is partly due to early studies suggesting that that they have a pro-tumoral activity due to the secretion of a cytokine very important, which is IL-10, as well as contrasting prognostic associations across cancer types that you can see here very nicely summarized on the right-hand side. And you can see depicted by colors that even in the same cancer type, you can see the divergence of association with clinical prognosis depending on the B cell subset or that you are actually exploring. And this really highlights the need for better technologies to be incorporated to actually understand what is the key elements of B cells. And this actually came in the last decade with a major shift in perspective coming from the discovery of tertiary lymphoid structures in and around tumors, which have been positively correlated with both patient survival and response to immuno-oncology therapies like PD-1 blockade across multiple cancer types. It started with actually ovarian melanoma and also in the context of CRC. These ectopic microstructures, which you see here in the multiplex immunofluorescence sections from this particular patient diagnosed with stage 2 from this Caucasian cohort, seems to resemble the secondary lymphoid follicles, frequently containing both B cells, as shown here in gray with germinal center B cells, and T cells in germinal center-like structures that are conformed with CXCL13, and these are features that strongly associate with favorable clinical outcome. In this particular cohort, albeit being rather small in terms of sample size, you can really see that after 2 or 3 years after diagnosis, those patients who have evidence of mature TLS near their tumors, they seem to recur much less than those patients with no or immature TLS in the biopsies. And this is consistent not only in primary tumors but also in metastatic cohorts. This is actually work from Jérôme Gallon in France, where he shows that where B cells are in close proximity with CD8 T cells, which are very important for cytotoxicity and lysis of malignant cells, are also associated with good prognosis. And these findings really indicate that B cells and also the environment that is supportive in tertiary lymphoid structures might be hubs of anti-tumor immunity worth exploring for improving vaccines. And this is where our collaboration with the Hospital General and the Instituto Politécnico Nacional with Oxford Cancer started to play, because our goal was to study how in real life, in patient cohorts, how lifestyle and impacts disease progression and its underlying immune response in the context, for instance, of tertiary lymphoid structures and B cells across the globe, to be able to gain insight as to potential candidates for cancer vaccines that are global or population specific, that are curtailable and available for people across the globe. Also, there's an urgent need to do this in Mexico City, given that CCR really occupies first place in deaths because of cancer, and also the alarming numbers of cases that we see as we hear from the medical team in Hospital General in terms of inflammatory intestinal conditions like Crohn's disease, colitis, and a lot of young people, especially that come to the hospitals with this diagnosis being suggested. And also the fact that the 30% of the cases, they do present with malnutrition. We felt that doing an alliance with Hospital General and together with Politécnico Nacional, which is really at the heart of Mexico City, provides an opportunity for us to understand better the immune response in the Mexican patients and Mexican population, that can give us information for better vaccine strategies and better treatments. So our mission is to seek out novel candidates for vaccines for high-risk populations and also improve treatments that are inclusive. And we hope to do this and reduce the slope of IBD and CRC cases in Mexico in the future, especially in young patients, by working closely together with our project partners, as well as influencing public policy and provide information to ensure translation with a multisectoral lens. As I mentioned, this strategic collaborative alliance between Oxford Cancer at the University of Oxford, the Instituto Politécnico Nacional, and the Hospital General aims to establish a cross-institutional network between UK and Mexican scientists to improve global health in general. Our vision in the context in Mexico specifically, we would like to propel Mexico towards cancer translational research together with numerous scientific disciplines, so partnering with people across disciplines in Mexico, including medicine, biology, engineering, and computer sciences, by building local research capacity and strengthening the partnerships that we have, not also with our academic partners, but also hopefully with government and industrial partners across Mexico and around the world. We think our strategy is led by a research question in terms of looking at these TLS hubs, and by having uniquely located cohorts and a multidisciplinary expertise of the team. We are excited about the opportunity to actually contribute to scientific findings in the future. So what do we know about Latin American patients so far? We're not the first. There's precedent on looking for Mexican genomics, and actually this came out as you know, as the cover of Nature a few years back. And this is actually beautiful work in collaboration with NDM Oxford and also Simbe. And also there's another work with the Mexico City Prospective Study with UNAM, and also NDM Oxford with Jonathan Emberson, and collectively we have dissect the whole genome sequencing of the Mexican population with the whole genome-wide association analysis, and also specifically in Mexico City by recruiting 140,000 adults that would follow longitudinally for more than 20 years. And in the context of, and this provided a lot of information as to our genetic footprint that we can learn and apply now in the next step in terms of cancer. What we know so far about the cancer association is it actually comes from a study from the US, which suggests that Hispanic patients had significantly higher odds of presenting with a pathogenic APC variant, which is very important in the context of CRC. And also MLH1 variants that versus Caucasian patients. And this might suggest as well, given their associations with a different immune response that is important to analyze. The Mexico Cancer Initiative actually came as a conversation with our director from Oxford Cancer, Professor Tim Elliott, and the director from the Politécnico Nacional, Arturo Reyes Sandoval, and trying to identify the needs and the potential collaborations that could harness both institutions. And this is where the idea of studying patients in the immune system in Mexico and the UK as a mirror cohorts, we felt that it provides a very important information, for instance, for the development of cancer vaccines, and can develop into the establishment of cancer biobanks for future research to strengthen our Mexican network in the future. And so our team and our network started to grow since 2022. And now I'm very delighted to say that the network really has expanded across Hospital General and also Instituto Politécnico Nacional. And we have now an established team that supports the Mexico Cancer Initiative as well. Together we have involved medical students, and we're now collecting samples from CRC patients who have been diagnosed, unfortunately, with CRC lately. We have successfully established core assays at Hospital General and IPN, and this actually stems from the collection and processing of samples from patients who have consented in the coloproctology unit at Hospital General to provide tumor and adjacent tissue sections, as well as paired blood samples and fecal samples. And together with a nutritional profile that will give us information as to the environmental factors that could be associated with the immune prognosis. And with this, we have processed a broad array based on the Oxford Cancer pipelines, on the way how we're collecting in Oxford, to have FFP blocks, fresh frozen blocks, cryopreserved live tissue, also flow cytometry on live cells. We have the nutritional profile, fecal samples, peripheral blood mononuclear cells, plasma, and serum to be able to incorporate all the assays. And this is just on the right, you can see a list of the not exhaustive core assays that we are currently establishing in Mexico City that will complement the work that we will do also here in Oxford. And this includes H&E analysis of these specific structures, 16S sequencing for microbiome analysis, metabolic profiling. We're working on a low-plex sequencing as well that can quickly provide information to the patients that we are to the research that is ongoing in Hospital General. And also, most recently, we established a Tedicolor spectral flow cytometry panel to look for immune cells in fresh tissue. And this it provides a really quick glance of what the immune system is doing at that particular time in these patients. And I just wanted to show you very briefly some of the assays that we are obtaining, that we have established in Oxford, and things that we are implementing as well within the Mexico initiative. On the left, you can see it's an example of a tumor tissue from a patient diagnosed with stage 3 colon cancer before treatment from the Combat Cancer Oxford Cancer Consortium. And you can see here is a 60-plex immunofluorescent gene panel that aims to look at the immune system in the tumor microenvironment. And you can clearly see here that in cyan, you can see the T cells infiltrating in this particular section. You have the B cell zones, proliferating B cells in white, and you have in pink the plasma cells that are important for antibody production. And you can see this is not very clear in this one, but actually the epithelial cells and malignant cells are actually on the right-hand side, suggesting antibody mediated cytotoxicity that could be happening, and we were already seeing in our preliminary data. So these type of assays really provide a very a look and a zooming into local antibody responses that could provide valuable information for the development of cancer vaccines that we're establishing at the moment. And this is, of course, complemented with high-dimensional flow cytometry panels that we have developed here in Oxford that comprise almost 120 proteins that we can look at in the single cell level across blood, tumor tissue, and adjacent tissue to really dissect the role for the immune responses in this particular cohorts. And this is followed by downstream validation functional assays that would eventually lead into candidate vaccines in the future. And today, I just wanted to give you a very quick example of the work that we have implemented specifically in Mexico City. So we decided to compress this 115 panel array that we had in Oxford into selecting the most key 30 markers that can give us a first look information on the immune system on the Mexican patients with CRC. And we are very excited that it has been very successfully established in collaboration with the Dr. Constantino Lopez Macias team at the Centro Médico Nacional Siglo XXI. And this includes a panel, what we call panel 0, that allows the identification of 20 and more immune cell subsets in terms of activation and function as well, that can loop into the analysis and becomes the data commons of these of the patients that can be then used for future analysis. So we think that we're very excited about this initiative, and also the scope and the network that it has created in the past years. And we feel by translating all the initiatives activity to maximize impact for the Mexican community while building local research capacity via the cancer biobanks that we're establishing to consolidate, hopefully in the long term, an initiative towards translation. And we're thinking that this specific research, most importantly by looking at TLS, could provide information for the development of future cancer vaccines that can be shared across either globally or can be curtailed for specific populations. So with that, I just would like to acknowledge all the people who have been involved in the work that has happened in University of Oxford in terms of the biology for TLS to try to understand these hubs. And also most recently, all the institutions, and especially also Hospital General, who have open their doors for the alliance for establishment of the Mexico Cancer Initiative. And we think that this is just the beginning. We are open for collaborations, and as you can see, we have all these core assays that we're more than happy to involve more people. So please join us in accelerating research and, most importantly, hope. And with that, I just would like to thank all the patients that have been consenting for our work here at the Mexico Cancer Initiative, and thank you for your attention.